we are now starting the actual process of photosynthesis. So when we say the process, this process is completed in two steps. One, which is a light dependent reaction, and the second is light independent reaction. The reaction which is dependent on light, that means light is essential for that reaction to take place, we call that process as light reaction, or it is also known as photophosphorylation. And the reason why it is known as photophosphorylation is, one, because it is taking place in presence of light, that is photo, and phosphorylation because this process is going to give us ATP synthesized, and ATP will be synthesized from ADP. That is, in diphosphate, when one more phosphorus gets added, that is what is phosphorylation. The light independent reaction is commonly called dark reaction. But this should not be confused with the thing that light independent means it is taking place in dark. This means that it can take place in presence of light as well as in absence of light. And this dark reaction, complete process, was explained by Kelvin. So it is also known as Kelvin cycle. And it is also known as C3 cycle. We'll see why we are calling it C3 later on. So we are starting with the first step, that is the photophosphorylation. And this photophosphorylation also takes place in two different pathways. So let us start with the first pathway, which is known as non-cyclic photophosphorylation. Non-cyclic photophosphorylation. This process is the main pathway which takes place, and in this we will get ATP as well as a reducing power synthesized. Let us first understand the process and simultaneously we'll write down certain important points. This process starts with PS2. We know PS2 is P680 nanometers. That means the reaction center chlorophyll A is going to absorb or absorbs 680 nanometers. And just to understand that there are other accessory pigments, we are also drawing these other pigments. So this is the photosystem that we are talking of. And in case of PS2, it has its primary electron acceptor. Primary electron acceptor. We will write down the abbreviations and their full forms on one side. So PEA is primary electron acceptor. This is the part of the photosystem. Now when sunlight falls on these pigments, what happens is chlorophyll A absorbs wavelength, accessory pigments also absorb their respective wavelengths, and they pass on these photons to the main, that is reaction center chlorophyll. A pair of electron gets excited and this pair is accepted by this electron acceptor, and this is the first one, that is why we are calling it primary electron acceptor. Now this electron, which is at a higher energy level, will come down the gradient through an electron transport chain. So this electron is going to come down. So from higher energy level to lower energy level. It passes through 3 electron acceptors. These electron acceptors are PQ. So let us write what PQ stands for here. It stands for plastoquinone. Then the next electron acceptor is written as Cy. This is actually cytochrome complex. And third is Pc, that is plastocyanin. So now these are the three electron acceptors. That means this pair of electron which got excited from chlorophyll A now comes from primary electron acceptor to plastoquinone. Then from plastoquinone it comes to cytochrome complex, and from cytochrome complex it comes to plastocyanin. Now when it is coming through cytochrome, it releases that extra energy, and that energy is used to combine ADP with inorganic phosphate, and ATP is synthesized. And that is why we have called this process phosphorylation. And because it is taking place with the help of the solar energy or light energy, photo word is added. So it is photo because this is the light energy, and with this light energy, the energy, chemical energy is synthesized. So we are converting, or the plants are converting solar energy into chemical energy in the form of ATP. Now let us see what happens to this pair of electron. This pair of electron from plastocyanin is actually going to PS1. So from PS2, the electron pair got excited. It was taken by primary electron acceptor. Then it went through an electron transport chain. And from this, now it is taken by, so let me write it here, PS1. So this electron goes to the reaction center chlorophyll of PS1. That is P700. This reaction center chlorophyll A absorbs 700 nanometers. So now this pair of electron has been sent or has been accepted by chlorophyll A of photosystem 1, and it also has those accessory pigments along with the reaction center. Same thing, the light energy is falling on this photosystem also. All pigments are absorbing their corresponding wavelengths, and they are passing these photons to the main chlorophyll. Again, this photosystem also has its primary electron acceptor. So when this chlorophyll A receives sufficient photons directly also and from the accessory pigments also, again a pair of electrons gets excited, and this pair is accepted by the primary electron acceptor. From here, this electron pair is sent to, again, a small electron transport chain which has FD as its electron acceptor. So this FD stands for ferridoxin. And then this electron pair is given to NADP molecule. So this electron pair is given to NADP. Now this electron pair, when goes to this NADP, it will get reduced. How that gets reduced? With that we will see in a minute. So what has happened so far is light energy is taken by PS2. So that means non-cyclic photophosphorylation starts with PS2. Both photosystems are participating. But first it is PS2 and then it is PS1. So a pair of electron gets excited, taken by primary electron acceptor, then it passes through an electron transport chain which is made up of 3 electron acceptors: plastoquinone, cytochrome, plastocyanin. When this electron comes down the gradient, it loses that extra energy at cytochrome. That energy is used to synthesize ATP. The pair of electron is now accepted by the reaction center chlorophyll of PS1. Again, by absorbing energy or photons, that electron pair gets excited, taken by primary electron acceptor of PS1, passes through a small electron transport chain and is given to NADP. So now the NADP has electron. Now we have to understand one thing, that if light continuously falls on this PS2, a pair of electron will continuously get released. So one pair of electron goes, then next pair goes, then next pair. So how long can the electrons be lost from this chlorophyll molecule? There has to be a mechanism to replace those lost electrons. This replacement is done by, let me write P680 here. This is done by a process which is called photolysis of water, where water splits in presence of sunlight, and the process is known as photolysis. It splits into a pair of electrons, a pair of protons, and one atom of oxygen. So we can write the atom of oxygen as just one O, or it can also be written as half O2, because this is the nascent oxygen and immediately it is going to bind with another nascent oxygen to form the molecule. But this electron which is generated by the photolysis of water, this electron is given to the chlorophyll A. So when light falls on this chlorophyll A, one pair of electron gets excited. By photolysis of water, that electron pair is replaced. That means that electron pair is given back to this chlorophyll A. So its loss is made up. The process is photolysis of water. This also takes place in presence of light, and that is why photo for light and lysis is for splitting. Now this oxygen which is given out here is actually the byproduct. When we say in photosynthesis carbohydrate is synthesized and oxygen is given out as a byproduct, this oxygen is coming from water. So water splits. So one, that is pair of electron, has been used up to replace the lost pair of electron from chlorophyll A of PS2. Oxygen is a byproduct. Now the protons remain. These protons, that means we are talking of this proton. These protons are given to NADP. So this proton pair is coming to NADP, and by taking this proton, this electron, NADP gets reduced to NADPH plus H plus. It can also be written as NADPH2. That means in this process we are getting two things. We are getting ATP and we are getting our reducing power which is required to reduce carbon dioxide in dark reaction or in Kelvin cycle. This complete mechanism, which is known as non-cyclic photophosphorylation, is also termed as Z scheme. And the reason term Z scheme is given because it goes like up, coming down and like this. Actually, it looks like an alphabet N, but probably they thought it starts from here, then comes down and then goes to PS1. So it is also known as Z scheme. Now the location. This non-cyclic photophosphorylation, because it requires photosystems or pigments, it takes place in thylakoids. And in this thylakoid membrane, the complex which helps in photolysis of water. So this is taking place in thylakoid. This is the location. And to be very specific, water splitting, that is the complex which helps in photolysis. Complex for photolysis of water is present on the inner membrane of thylakoid. Inner membrane of thylakoid. And the enzyme which is required here, this enzyme which is needed here is actually NADP reductase. So this NADP reductase, it is present on the outer surface of thylakoid. So NADP reductase is on the outer membrane of thylakoid. That means if we draw a small thylakoid here, the complex which is going to help in photolysis of water is here, and NADP reductase is going to be on the outer side. Now this location is required to understand where water molecule will split and where NADPH or NADPH2 will be synthesized. The complex is on the inner side. That means splitting will take place here, that is in the lumen. So oxygen will be produced in the lumen of the thylakoids. And because this enzyme is present on the outer side, this reduction is going to take place here. So this NADPH2 will be synthesized in the stroma. Again, we will be talking about this in the process of chemiosmosis, where we actually understand how ATP is synthesized. But just a quick recap of the process of non-cyclic photophosphorylation, which is one of the pathways of light reaction or light dependent reaction, also known as Z scheme because of that shape of the pathway. It starts with PS2. PS1 also helps. PS2 is associated with photolysis of water. The reason is when light energy is trapped by chlorophyll molecules, a pair of electrons escapes chlorophyll. To replace that pair of electrons, photolysis of water takes place. The proton which is released during this photolysis is used to obtain the reducing power. And third thing, that is oxygen which is given out, is the byproduct of photosynthesis. The pair of electron is accepted by the primary electron acceptor of PS2. Then it passes through an electron transport chain. There are three proteins or three complexes which act as electron acceptors. The sequence is plastoquinone, then cytochrome, then plastocyanine. Then the pair of electron goes to PS1, again gets excited, passes through ferredoxin and is finally given to NADP. So NADP gets electrons as well as protons which are coming from splitting of water, and it gets reduced. So in this process, that is non-cyclic photophosphorylation, what all things are being obtained? ATP, reducing power, as well as this oxygen, though it is a byproduct, but it is synthesized or produced during non-cyclic photophosphorylation, and this is taking place in the thylakoid membrane. So this is one. Now next is going to be the cyclic photophosphorylation. This is predominant because for dark reaction two things are required. ATP is also required and NADPH2 is also required. The next process, which is cyclic photophosphorylation, that gives us only ATP. So this is the major pathway, and cyclic photophosphorylation also takes place, but less as compared to this one. Now the last thing, we know why it is known as photophosphorylation, because ATP is synthesized in presence of light. We need to also understand why is it known as non-cyclic. The reason for calling it a non-cyclic pathway is that the electrons which are coming from chlorophyll, forget about this photolysis, the electrons which are coming from chlorophyll A, they never come back to chlorophyll A of PS2. In cyclic, we will see that the electron which are released from that chlorophyll will come back to chlorophyll molecule, and that is why we call that process cyclic. And here the electrons of chlorophyll A, once they are released, they are finally going to NADP. They never come back to chlorophyll A again of PS2. We are replacing it from or recharging it from photolysis of water. And once we take cyclic, that thing will be more clear to us. So let us now talk about cyclic photophosphorite.